library of various vancomycin and resistant enterococci. They ran vancomycin resistant enterococcus strains and prepared a library just like the one in SARS-CoV-2. So they prepared a library and they taught a machine program with the help of a convolutional neural network to recognize those patterns in normal or clinical isolates. So the multi-top machine was actually only able to identify whether the actual organism was enterococcus or not. But feeding those data and those spectra into the machine learning software with the help of the CNN, it was actually able to identify those particular strains of enterococci which were vancomycin resistant. So direct detection of resistance from the isolate without application of any further tests, whether it is phenotypic or otherwise. This is actually beneficial to the patient, not just to the patient, but also from an epidemiological point of view. That is, application of proper infection prevention and control practices were rapidly done, prevention of an outbreak, prevention of spread of those infections, and of course, reduction in the hospital costs, reduction in stay in the hospital. All those secondary benefits are also obtained from it. So these three important things which are being done with the help of Monitor, which are separate from actual microbial identification, whether it be bacteria, fungi, or microbacteria. This is the application which is being done in clinical practice or in laboratory practice these days. So this is quite promising. So can we do more? Answer is of course yes. We can actually increase our collaborations with different kinds of fields, in, for example, in biotechnology, with machine learning, with AI scientists and all, to develop better and better algorithms, better and better machine learning patterns. Because Marditov is actually a very, very powerful tool. It can detect any kind of proteins. It's what we do with that understanding of proteins is what it does. So, what has been proposed, what has been tried over the world over? So, that since the study about vancomycin resistant enterococci was quite successful, other studies have also been done in the respect of gram-negative bacilli also. This one study from Switzerland, they tried to generate such kinds of databases about the resistance patterns in gram-negative bacilli also, like for example Pseudomonas acinetobacter and so forth, which are more commonly found in hospital settings. So, this particular study or this particular approach is currently not that much in vogue. The reason for that is one kind of genotype or one kind of resistance gene can manifest in many other patterns. So, encompassing all those patterns, all those phenotypes and developing spectra out of them is quite a challenging task. And it requires much more application of machine learning and AI development, AI software. So that's why it's something in progress right now, but it shows quite a bit of promise. Now, another thing which is a bit more further down the road in cancer is in cancer detections. So there are two important technologies which I want to discuss over here. The first one is something known as multi-high-plex. So multi-high-plex is technology that they are combining with immunohistochemistry for detection of anaplastic or metaplastic cells from FNAC samples, that is fine needle aspiration cytology samples, which are liquid in nature. So from liquid samples, they are able to generate a pixel by pixel image, that is a dot which represents a cell. So one cell in one particular color is going to signify a normal cell. A cell in a different color, a dot of different color will signify an anaplastic cell or some or the other kind of mutation is there. So this can give a direct pictorial representation of what is actually happening inside that sample, which can actually help us or help the clinician to determine whether the patient requires further follow-up about cancers or not. So what it does, this kind of technology, it reduces the time for detection of such anaplastic or mutated cells by 4 times as compared to immunohistochemistry. So the crux of the whole matter, the crux of everything which has to do with Marditov is actually saving of time. Right? So the next technology I'd like to discuss here is something known as multi-MSI. So MSI stands for mass spectrometry imaging. So the previous technology, it could generate pixelated versions of tissue samples or rather liquid samples. So this technology is going to help us generate such images, actual pictorial or actual images of the tissue which is there or the proteins of the tissue which is there from actual biopsy samples. As you can see in the image over here, it is a multi-MSI image of topoisomerase 2 inhibitor in a colon cancer in a mouse model. So these highlighted regions in purple color, they are actually the anaplastic cells which are supposed to be found in the colorectal cancer. So this was done in mouse models. Human testing has not been done yet, so we don't know whether it will work or not. So whether or not it has if developed successfully enough, if proper machine learning integration has been done, would it be able to replace histopathological examination? It's something which we could ponder. Because histopathological examination requires so much of expertise, so much of time, so much of reagents and all that. This is quite simpler in comparison. Right, so now detection of cancers is dependent on various types of omics. Like for example, starting with genomics, which actually has to do with the nuclear or the genes of the person of the cell, followed by transcriptomics, proteomics, metabolomics, and all those. So all these together, combined together, they are going to form certain biomarkers, which are going to ultimately help us in diagnosis of cancers. So MonData actually deals with the proteomic biomarkers. So basically, what I want to emphasize upon is that MonData detects proteins. MonData is fast. MonData is useful in detection of AMR, in surveillance of AMR, and taking actual evidence-based steps in epidemiological prevention of such a spread of such genes. And also it has a lot of strong future in cancer diagnostics. So I'd like all of you to just think over this. Since the topic of the theme of this conference is microscopes to genomics. So talking about genomics, we can say that genes which encode for certain characteristics, they may or may not get expressed. We don't know what is the dominant, what is the recessive. But proteins which are there in the cells, they are actually the expressed genes. Now since Monday detects proteins, what do you want to believe? Do you want to believe what can be expressed or not expressed, or what has actually been expressed? I'd like you to think over this. Thank you.
Thank you, Dr. Akshay. First of all, for sticking to the time and for sharing the future applications of the wide topic diagnostic microbiology. Dr. Akshay Karyakarte has some big shoes to fill, and we know he will be up to the challenge. Thank you so much. Felicitate all our speakers at the end of the session. After this, I call upon Dr. Rashmita Das. She is the next speaker. Since Rashmita Das has joined the PG Government Medical College after her MD, she has exclusively worked in molecular microbiology. Dr. Rashmita Das is a dedicated clinical microbiologist with over 5 years of experience in research and diagnostics. After earning her master's degree in microbiology from Grant Medical College and Sir J. J. Group of Hospitals, Mumbai in 2019, she has been associated with Department of Microbiology at BGGMC Pune. Dr. Das has played a pivotal role in National COVID-19 RT-PCR laboratory and has extensive expertise in next-generation sequencing, having sequenced thousands of SARS-CoV-2 samples under the leadership of Dr. Rajesh Karyakarte. She was instrumental in establishing the Center of Excellence for Genomics at BJGMC in 2022. She is actively involved in genomic surveillance as a part of INSAPOW and collaborates with several prestigious institutions. Dr. Das is committed to advancing the role of genomics in understanding and diagnosing the infectious diseases with expertise spanning molecular diagnostics and genomic research. Now I hand it over to Dr. Rashmita Das.
Thank you, madam. A very good morning to everyone. It's an honor to be here today to talk about a topic that is reshaping the landscape of healthcare and public health, that is genomics. In the next few minutes, I'll be exploring the multifaceted role of genomics in transforming medicine. I'll begin by discussing how genomics has revolutionized diagnostics. It has strengthened it will strengthen the pandemic preparedness, driving innovations in vaccine development and is pioneering in therapeutic breakthroughs. So to start with, my first Heading would be pathogen identification and discovery in the age of next-gen sequencing. The traditional diagnostic methods often require multiple tests that are hypothesis-driven. On the other hand, with metagenomic next-generation sequencing, we can now detect all microbial DNA or RNA in a sample in a single run. This hypothesis-free approach has been pivotal in identifying co-infections, novel pathogens, and has been helpful in understanding the full microbial landscape. One of the most compelling examples of metagenomics utility was the discovery of SARS-CoV-2 virus in Wuhan in 2019. This breakthrough was instrumental in determining the cause of the mysterious pneumonia outbreak in late 2019, highlighting the technology's critical role in outbreak response and public health. But SARS-CoV-2 is just one example. What about other unknown threats that genomics can help us uncover? So studies have suggested that over 3 lakh mammalian viruses remain undiscovered, many of which pose zoonotic threats. By integrating genomics with machine learning, scientists have already uncovered 1,60,000 new RNA viruses, some from extreme ecological niches. The ability to identify these threats early is crucial for preventing large-scale outbreaks. Now again, also historically, bacteria were classified based on their morphology, growth requirements, and pathogenic potential. And this approach